Chapter 36, Resource Acquisition and Transport in Vascular Plants. So this is looking up from the bottom of giant redwood trees. So vascular plants, by vascular, remember from last chapter we talked about xylem and phloem. So the evolutionary journey onto land involved differentiation into roots and shoots. Vascular tissue, xylem and phloem, transports its nutrients to the plant, and such transport may occur over long distances like these giant redwood trees. Okay, so transport in vascular plants occurs on three scales. The transport of water and solutes by individual cells such as root hairs. Remember, root hairs are the tiny little, practically microscopic hairs on roots that increase the surface area of roots. Short distance transport of substances from cell to cell at the levels of tissues and organs, and we'll talk about that, how they go through gap junctions or symplastic or apoplast outside the gap junctions, but in the cell walls and extracellular spaces. And then long-distance transport within the xylem and phloem at the level of the whole plant, from root to the very top stem. So transport in plants, a variety of physical processes are involved in the different types of transport. So here, water leaving through the stomata, this is transpiration. Loss of water from leaves creates a force within leaves that pulls the xylem upward. Okay? So this is water and minerals in the soil are absorbed by roots. Water and minerals are transported upward from the roots to the shoots as xylem sap and then out through transpiration. And then roots down here also exchange gases with air spaces in soil. That's why a plant will die if it's in waterlogged soil, because it can't get any oxygen. So taking in oxygen, discharging CO2, and here the phloem, sap can flow in both ways, okay? Here it just looks down, but it actually can flow both ways between the shoots and roots. It moves from sites of sugar production up here in the leaves or storage in the roots, okay, to sites of sugar use or storage. So light is needed for photosynthesis. Sugars are produced by photosynthesis in the leaves, and through stomata in the leaves, CO2 is taken in to fix carbon for photosynthesis, and oxygen is released as a by-photosynthesis. Okay, we talked about proton pumps a little bit in Bio-101, but proton pumps in plant cells create a hydrogen ion gradient. Okay, so active transport, ATP needed to pump these hydrogen ions out. So create a hydrogen ion gradient that is a form of potential energy and can be harnessed to do work. So this voltage is known as membrane potential. So negative and then positive over here. So proton pumps. This is a membrane potential and cation uptake. So the cations, potassium here, for example, are driven into the cell by the membrane potential, okay, because this is positive and negative, so going through this carrier plane. Here, co-transport of an anion with hydrogen, okay, so cells accumulate ions, here nitrate, for example, by coupling their transport to the inward diffusion of the hydrogen ions through a co-transporter. So this takes in the nitrate along with the hydrogen. Co-transport of anions. These are anions because they're negative. And then lastly, we talked about this first semester, co-transport of a neutral solute such as sugar, okay, sucrose. Plant cells can also accumulate a neutral solute such as sucrose, which are the triangle S's, by co-transporting hydrogen ions down the steep proton gradient. Okay, so they are pumped out and then they flow back in down the gradient, taking sucrose with them. Okay, effects of differences in water potential. So, common sense, plants have to balance water uptake and water loss. So osmosis definitely comes into play and determines the net uptake of water or water loss by a cell affected by the concentration of the solutes in the water and then the water pressure. So water potential is a measurement that combines the effects of solute concentration and pressure. So water potential we're going to be talking about. Water potential determines the direction of movement of water, and it's important to remember this. Water flows from regions of higher water potential to regions of lower water potential. So the roots have higher water potential, flows to lower water potential, which is up in the apstem. Both pressure and solute concentration affect the water potential. So the solute potential of a solution, okay, again a little review, is proportional to the number of dissolved molecules. The greater the number of dissolved molecules, the greater the solute potential. Pressure potential is the physical pressure on the solution. So, quantitative analysis of water potential. So here, the addition of solutes reduces water potential. Here is pure water. Down here, 0. Addition of solutes, and it becomes a 0.1 molar solution. The water potential has now been reduced below 0 to negative 0.23. So this U-shaped apparatus, a membrane separates pure water on the left from a 0.1 solution on the right, containing a solute that cannot freely pass across the membrane. So these different values in the left and right arms of the U2 are given for initial conditions before any net movement. Okay? So if no pressure is applied, this right here determines net movement of water. So here, positive pressure increased here, this symbol right here, on the right raises the water potential on the right, here making the same in both arms. Okay, so it's the same, so eventually there is no water movement. Negative pressure reduces water potential. In this case, negative pressure on the left decreases the pressure on the left, causing net water movement to the left. Okay? So here, 0.3, 0.2. Gain water and become turgid. So here, there's a net uptake of water by osmosis, causing the cell to become turgid. With this tendency for water to enter is offset by the back pressure of this strong cell wall here. Okay? Water potentials are equal for the cell and its surroundings. Okay, so this is the same, and this will make plant revive again. Okay, so turgor loss in plants causes wilting, which can be reversed when this impatience is uttered. Transport is also regulated by the compartmental structure of plant cells. The plasma membrane directly controls the traffic of molecules. Okay, plasma membrane inside the cell wall into and out of the protoplast, the inside of the cell. So the plasma membrane is a barrier between two major compartments, the cell wall and cytosol. Okay, so the third major compartment in most mature plant cells is the vacuole, which takes up a lot of the inside of the cell. It's a large organelle, can occupy as much as 90% of the cell. The vacuolar membrane regulates transport between the cytosol and the vacuole. Okay, so here are cells, the cell wall. Here are the plasmodesmata that link the cytosol, plasma membrane. Here's the tonoplast around the vacuole, and water moves back and forth. So this is a apoplast up here moving through the cell wall. Moving within the cytosol is a symplast. So an apoplast is the continuum of cell walls and extracellular spaces. So solutes and water can move through there. And a symplast is the continuum of cytosol connected by plasmodesmata. So in most plant tissues, the cell walls and cytosol are continuous. The cytoplasmic continuum is called the symplast, remember that, and the apoplast is the continuum of cell walls and extracellular spaces. So transmembrane route, water can travel out of one cell across a cell wall and into another cell. Or water and minerals can travel via the continuum of the cytosol through plasmodesmata, or water and minerals can travel the apoplastic route via the cell walls and intracellular spaces. So in bulk flow, movement of fluid in the xylem and phloem is driven by pressure differences at opposite ends of the xylem vessels and sieve tubes. So remember, pressure differences are greater to lower pressure. So water and mineral salts from the soil enter the plant through the epidermis of the roots. So here is a cross-section of the root, epidermis, root hair. This is the epidermis. And ultimately flow through the root system. So here, this is the apoplastic root. Uptake of soil solution by the hydrophilic water-loving walls of root hairs provides access to the apoplast. Okay, through here. This is the apoplastic root. You can see extracellular through cell walls. Okay. Water and minerals can then diffuse into the cortex along the matrix of walls. Okay, this is the symplastic root here, the blue line. Okay, see it's going through the plasmodesmata. Minerals and water that cross the plasma membranes of root hairs can enter the symplast. And then the transmembrane root here. As soil solution moves along the apoplast, some water and minerals are transported into the protoplast of cells of the epidermis and cortex, and then they can move through the symplastic root. Okay? So here, right through here, the endodermis controls entry to the stele. Okay, so the stele are the vascular vessels. Okay, the vascular cylinder here is the endodermis around that, this area here. Okay, so it controls within the transverse and radial walls of each endodermal cell is this Caspian strip. Okay. Okay, this is a control point, the Caspian strip. It's waxy material, blocks the passage of water and dissolved minerals. Only minerals already in the symplast or entering that pathway by crossing the plasma membrane can detour around the Caspian strip and pass into the steel. Okay? So they can detour around that.